Sounds good. Good afternoon, everyone. My name is Jeremy Barr, and I'm currently working out at the Roa Lab at San Diego State University. And I'm going to talk to you today about our recently proposed bacteriophage adherence to mucus, or BAM model, and the role that this plays in mucosal immunity. So a lot of this work has just recently been published in PNAS, so I'm not going to cover a lot of the background. We also have a poster up, so you can come and talk to me about that data there. And we also just have a recent bacteriophage article just out in the last couple of weeks. You can get some more background information there as well. But I'm going to cover some of the basics of what we've shown with this BAM model. So we're proposing here that mucosal surfaces accumulate phage. And the phage accumulate to these mucosal surfaces via these capsid-displayed immunoglobulin-like domains, or Ig-like domains, that form weak interactions with glycan residues present within these mucus surfaces. And it's actually this accumulation of phage that forms this antibacterial layer that both reduces bacterial attachment and colonization of the mucus and subsequently protects the underlying host epithelial cells from cell death and infection. So it shouldn't surprise you to learn that mucus is really everywhere, and there's a huge array of animals that utilize mucus and its protective benefits. So the BAM model proposes this as a ubiquitous, non-host derived layer of immunity that's mediated via phage accumulation to mucus. And it also proposes or demonstrates a novel symbiotic relationship between phage and metazoan hosts, whereby phage benefit through increased interaction rates and replication with bacteria and mucosal surfaces, and metazoan host benefit through decreased bacterial load and subsequent pathology of their mucosal surface. So a lot of the work that we did in this paper was actually done using in vitro tissue culture cell lines that produce a very basic mucosal surface. So we're using these mucus tissue culture cells and phage T4 as a model system, and some of the results we show were that these mucus adherent phage actually protected these tissue culture epithelium cells from bacterial insult and reduced their cell death. So a little bit about T4, phage T4, which we use for our model system. Phage T4 is actually known to encode 3 immunoglobulin-like or Ig-like domains. And it encodes these via this protein capsid known as the highly immunogenic outer capsid protein, or HOC. And the T4 structure was published a fair few years ago and was actually found to contain 155 copies of this HOC domain physically protruding out and coding this entire T4 capsid, with each one of these HOC domains coding for 3 putative Ig folds. So we subsequently showed that the HOC domain, well, one of the roles of the HOC domain is that it mediates this phage adherence to mucus. So knowing this, we have our wild-type T4 phage on the left here, and you can see all of these HOC domains protruding out from that capsid surface, encoding those Ig-like folds. And we obtained a HOC knockout mutant, and you can see that these domains are absent from this phage. And it's important to note here that these two phage can both infect and kill E. coli perfectly well under laboratory conditions, so there's no difference in infectivity via the loss of these HOC domains. So using these two phage, one of the first things we wanted to do was actually track the diffusion of both phage types in different mucus and buffer solutions. So we utilized a technique known as multiple particle tracking to do this. And basically what I'm showing you here is the effective diffusivity of these phage particles in a buffer and mucus solution. So if we look here, and this is the wild-type T4 here, so if we look at the wild-type T4 in buffer, you can see that it's moving extremely rapidly, and it has this flat diffusion profile over a 1-second time frame. And so this is indicating that the phage is not being hindered or slowed in this buffer solution over time. Compared to the same phage in a 1% mucus solution, and you can see the diffusion instantly drops, and we have this downward slope in diffusion profile over time, indicating that this phage is either sticking to or being hindered by that mucus solution. So if we do the exact same experiment with our HOC knockout phage, we can see that it's almost diffusing as rapidly in the mucus solution as it is in that buffer control. So this very strongly indicates that these HOC domains are actually required to mediate this phage adherence within that mucosal surface, or within mucus. So it's really important to note here that this phage adherence to mucus is not absolute. And by that I mean it's not that the phage is sticking to the first mucus molecule they see and not moving. Rather, it's this sort of aggregation, accumulation of hundreds of weak interactions between these Ig-like folds and the glycan residues present within mucus that enacts to maintain these phage within these mucosal surfaces over time. So this turns out to be really important actually, because we need to consider mucus secretion dynamics when we're actually looking at the effects in these systems. So mucus is constantly being produced and secreted by these epithelial cells, and it forms this sort of ever outwardly expanding mucosal layer that eventually reaches the top of the environment and is sloughed off and removed from that system. And so this is really important because phage must be able to diffuse throughout this mucus layer in order to mediate these protective effects. And in fact, if we had these phage sticking absolutely to the first mucus molecule they encountered, you'd have all the phage accumulating at the top, and they'd be the first to be removed from this system. So one of the side effects that we're noticing of this is it actually suggests that there's a balance between the strength and type of phage adherence to different mucosal surfaces and the interaction with that to the rate and turnover of mucus in different organisms. Or in other words, we think that specific phage are employing specific domains within an animal's mucosa that's quite accurately tuned to their secretion dynamics to maintain their adherence over time. So taking this all together, we wanted to take our quantitative diffusion profile data and actually apply it to do some modeling and track the accumulation and washout dynamics of phage in a theoretical mucosal surface. So to do this, we performed some in silico modeling. And here what we're doing is we're modeling the wash of a phage concentration at 10 to the 7 particles per mil across a mucosal surface of 100 micrometers in depth. And what we actually find is that quite rapidly you have an accumulation of both the wild type and the Hock knockout phage. Over a period of about 20 to 30 minutes, they hit this steady state accumulation. But you do notice roughly a 100-fold increase in the phage, and there is a preferential increase in those wild type phage sticking to that mucosal surface. So next we wanted to model the in silico washout dynamics of these phage. So we removed the phage washing across this surface and simply wash with a buffer control. And what we found was that, again, both phage actually wash out from this mucosal surface quite rapidly, and we have this preferential removal of the Hock knockout phage, which are diffusing out a lot quicker, whereas the wild type phage are maintaining within that mucosal surface at a longer rate. So the big question now is, can we actually show this in vitro, and can we see a differential effect between these two phage types? And it turns out that this is actually very difficult to test. And one of the reasons it's really difficult to test is the fact that we were still using these static tissue culture plates. And these plates don't accurately simulate these mucus secretion dynamics. And it turns out we actually require a consistent flow across these mucosal surfaces in order to mediate these washout effects. So we've now moved towards these microfluidic based devices. And this is one of these microfluidic devices that we've termed a BAM chip. And it's connected with tubing to a syringe pump driven system that feeds tissue culture media or growth media across those cell layers at a steady rate. And so this is a close-up of the chip, and it has a single channel attached to a glass slide with an inlet and an outlet port to maintain this fluid flow. And we're actually able to attach and grow mucus epithelial cells into these chips on the glass slide. And over the period of about a week, we can form these fully confluent mucosal epithelial cells that do start to somewhat differentiate due to these shear force dynamics. And this turns out to be really important because it allows us to actually infect these chips with both bacteria and phage at set concentrations and flow rates. And more importantly, it forces these bacteria and phage to actually attach and force their growth within that mucosal layer. And essentially anything that doesn't stick to this mucus is washed out and removed from this system. So we're eliminating all the growth factors up in the surrounding aqueous environment. So using this, we then wanted to test the protection of these wild type and Hock knockout phage in vitro using these BAM chips. So here are some of these BAM chips set up in multiplex in our incubator, and we've been pre-treating the surface of these chips with both wild type and T4 phage, again based off our in silico modeling dynamics to make sure that we hit steady state accumulation. And then we apply, or based off our in silico washers, we wanted to wash out these phage. However, from both our modeling results and some of our preliminary experimental results, we were noticing that we had very, very few phage actually maintaining within these mucosal surfaces over time. And so it raises the point, well, how can we mediate these protective effects if we're washing out such a large percentage of these phage? And it turns out that we somewhat answered this question. And we did this using the use of replication negative phage, which are these amber mutant 4344 phage. And so what I'm showing you here is an experiment with a replication negative phage. This phage can absorb, infect, and cause lysis from without of an E. coli cell, but doesn't produce any subsequent virion particles. And so what we see here with the replication negative, this is just our no phage control. So we see the bacteria very happily attach and grow in these mucosal epitheliums. And when we pre-treat with these replication negative phage, we see that there's no significant reduction in the number of bacteria on that mucosal surface, and we have very low virion production. compared to the same experiment with our wild-type phage that can infect and replicate, and we have a significant reduction in the number of bacteria, and we also have significant virion production. So this suggests that we can actually maintain, or we only require a very low number of phage within these mucosal surfaces to actually enable these protective effects, which is caused by phage replication and amplification. Or in other words, we only require a very low level of resident phage immunity, phage present within that mucosal surface to mediate that protective immunity. And this becomes important when we start to consider in vivo phage diversity. So if you imagine a mucosal surface with a large number of diverse phage, it only requires one infective phage to attack and kill a pathogen to enable this rapid amplification and protection. So knowing this, we wanted to put this all into our BAM chips and try and show this difference. So we now applied our in vitro washes, and again, we were based off our modeling attempts, and we're trying to wash out our Hoc negative phage, preferentially over our wild type T4 phage. And then we infected the chips with E. coli for the period of about 3 days. These were relatively long-term experiments set up within these chips. And so what we see is, these are just our control experiments, so this is just phage on its own after the washes, and we see we have relatively low numbers of T4, this is wild type shown here. We couldn't actually detect the Hoc knockout in this one experiment, so they were washing out preferentially, and just E. coli on its own, it's able to happily infect and grow within these chips. However, when we pre-treat the chips with T4 phage and then infect with E. coli, this is again the wild type, we have significant virion production maintaining in that mucus, and we have a big decrease in the number of attached and colonizing bacteria within those chips. Compared to the Hoc knockout phage, where we have reduced number of viruses present within the mucus, and again an increased number of bacteria there. So this really suggests that these Hoc domains are maintaining that phage adherence in mucus, and that there is some protective benefit over time within these in vitro systems. Now this is in vitro, and I have a lot of people come up and ask us where's the in vivo results, so I am pretty happy to show you some of the first in vivo experiments that we've done using these two phage types. So what we're using here for our in vivo test is again just testing the protective effects of our wild type T4 and our Hoc knockout T4 phage in the mouse vaginal tract. And so a really basic experimental design for our setup here. We pre-treat the mice with both the wild type and the Hoc knockout phage. We then wash the vaginal tract and then challenge it with E. coli. And then we're quantifying from this the PFUs and the CFUs of these phage at 1 hour and 18-hour time points. Now, there's a number of caveats for this experiment. E. coli is not normally present within the mouse vaginal tract normal flora. We do try and pre-screen the mice to make sure we're not selecting ones that do have E. coli present. We have added antibiotic selection pressures to our E. coli host. This may affect growth dynamics. And there could be any number of competing commensal phages that we're not aware of at the moment. However, we still did see some pretty exciting preliminary results. So what I'm showing you here are PFU counts and CFU counts over both a 1 hour and 18-hour time point of our T4 wild type phage. And so what we see in the PFU counts is that there's a very slight reduction over that 18-hour time point, compared to the CFU counts, where we have a relatively significant reduction in the number of bacteria that are maintaining there. Compared to our Hoc knockout phage, where we have a greater decrease in the number of phage that are maintaining within that vaginal tract, especially when you compare it to the 18-hour time point of the wild type. And with the, in the CFUs, you have a bit of an increase in the number of bacteria that are colonizing, again compared to the 18-hour time point of the wild type phage there. Compared to our SM buffer control, where we obviously detected no PFUs, and we actually had an increase in the number of phage that are growing within that mouse vaginal tract. So again, very preliminary, but it does suggest that these domains can mediate the maintenance of the phage within that mucosal epithelium over time, and that there may be protective antimicrobial effects as a result of this. So to wrap up, I'd just like to finish with some acknowledgements. I'd like to thank all my students that have helped out on this project and a number of other projects we've got going on in the lab. Big thanks to the Duran lab, who did a lot of the mouse work that I presented, the Pogliano lab at UCSD, who helped with the multiple particle tracking, and a big thanks to the SDSU Biomath group, who did a lot of the in silico modeling work that I showed you. And finally, a big thanks to Forrest. He unfortunately couldn't be here today. He's off in a cruise up in the Arctic, but he's been a great boss and mentor on this. So with that, I'll finish up and I'll be happy to take any questions. Thank you.
So it seems to me that phage would evolve to do this regardless of the effect of the bacterium. So it could wipe out commensal or beneficial bacteria too.
Yes, so roughly we know that about 25% of sequence phage have these domains. So there is a huge evolution factor here. But if your bacterial host never associated with mucus, then there'd be a really strong negative selection pressure for you to get stuck in that mucosal layer, and you wouldn't ever really replicate. So we think that really only phage that have their hosts somewhat associated with these layers probably express these domains. But there's definitely an effect on commensal bacteria that we're only just starting to look into. So.
Hi, is this on? Hello? Yes, certainly cervical mucus is negatively charged. I presume gastral mucus is too. Okay, so the question really is, have you shown that it's the immunodominant groups or whatever, these three groups on the hawk that are responsible for some kind of binding, or just the fact that these are positively charged regions of the hawk protein?
Yeah.
And I guess I didn't see any data to that effect, but,
sure.
but I guess the simplest question would be, are there regions of basic amino acids that might then be used to bind to the
So that was
mucus?
one of our first hypotheses. We thought this could be electrostatic interactions, and there potentially is a very small role that they play, but we have shown that these Hock domains are responsible for binding to sugars, and that was irrespective of mucus. And then we've done a lot of knockout experiments with different phage mutants to show this. And the poster that I presented has all that data, so I can run that through with you if you want.
Thank you.
Cool.